Hey, and welcome back again. So we're in this section on cells, and we've had a pretty detailed conversation so far. In this part, so that it doesn't end up being like an hour long, which you'll never watch, there's a couple of spots where I'm just going to direct you to reading some stuff in your textbooks, which you should be doing for everything we're talking about, but you should focus especially on these areas when they come up. And the reason I'm going to do that is simply because it's for space reasons. I don't want to spend hours and hours just sitting here talking at you, so I'm trying to keep these short, and so this is one of the ways I'm going to do it. So we're going to start getting into the cell, and we're going to start looking at some specific structures. The first thing we're going to look at is the cytosol. We'll look at that in a moment, and then we're going to look at the structure and function of the cytoskeleton. We'll talk about that a little bit when we get there. The flagella and the cilia. I want you to focus on the structure and the function of the flagella and cilia on your own as part of your readings, so that I don't add an extra 20 minutes to this recording. I'll talk about the endomembrane system because that is a little bit complicated and a little bit of explanation is good. And then the last two items on here, the mitochondria and the chloroplast, I think these are so heavily taught in high schools as part of biology, and I think people have got such a good understanding of what the mitochondria and the chloroplast do. Again, I'm just going to ask you to read about that and to think about the relationship between structure and function when you look at the mitochondria. So let's get cracking. Let's start off with the cytosol. The cytosol in the diagram here, the cytosol is this kind of brown with a little bit of a gradient on its material that you find in animal cells. Obviously you find it in bacteria too, and archaeobacteria, and down here in a plant cell again it's the brown substance. That is the thick gelatinous, very, very concentrated water-based material that's between the plasma membrane and then the membranes of any internal organelles, and it's also got structures like the centrioles supported in it. So that's what it is. It contains a lot of solutes, so it's got dissolved sugars, dissolved amino acids, it's got proteins, some of which are soluble, some of which are insoluble, and so it's very dense, and it's got numerous functions. And this is probably one of the problems with talking about the cytosol, is because it has so many functions, and so that's difficult to distill down into exactly what it does when it does so many things. So one thing we can definitely discreetly talk about though is the process of catabolism. That's the process of breaking molecules down. Now we talked about hydrolysis extensively already, and so if you were going to break down something like glycogen, then glycogen catabolism could be occurring in the cytosol. And certainly if we're looking at the oxidation, the breakdown of glucose by glycolysis, that's the first step in cellular respiration, that type of catabolic process occurs in the cytosol. Now, I'm not saying that only catabolic processes occur in the cytosol. That's not true. Catabolic processes occur in numerous other compartments in the cell, but you certainly find a lot of molecules being broken down. Now, that being said, you also find a lot of molecules being built up. So there's a lot of processes which actually work to synthesize molecules and build complexity in some of those organic polymers we looked at. Let's jump down to translation, because translation is a form of anabolic process. It's linking amino acids together, remember, using condensation chemistry, and so building those organic polymers, building that chain of amino acids into a polypeptide, which then folds up through those 4 levels of protein structure: primary, secondary, tertiary, and quaternary, to give us a fully functional protein. So translation is occurring in the cytoplasm. It's occurring elsewhere as well, and we'll talk about where else it's occurring, and you'll read about where else it's occurring as you're working with your textbook. Coordination. Now, what does that mean? What I mean by coordination is that cells receive information from their environment, and so they're constantly being bombarded with molecules which contain information. When I say they contain information, what I mean is they tell the cell to change its behavior in some way. And so when all these signals are being received by the cell, all of those different inputs are integrated in, generally to start with, the cytoplasm, and that's where a response to that information will be elicited. So a lot of signals and a lot of processes are coordinated in the cytoplasm. Touched on translation really quickly there, and the last thing to really mention here is that you're going to find a large amount of the cytoskeleton, a large proportion of the cytoskeleton is going to be found within the cytosol. So that's a little bit about the cytosol. It does tons of things. You can supplement what I've said here with some readings. Now let's move on and talk about the cytoskeleton. This is going to be a lot of information, and so it's not particularly complicated. It's just a lot of stuff. So this is a lot of information you've got to manage. So you want to be working on structure, on strategies to actually manage this amount of information. Lists are good, bulleted lists, that kind of thing. And this kind of stuff, to be honest, flashcards are good for this kind of stuff because it's just simple. You got to remember some stuff. Okay, what is the cytoskeleton? The cytoskeleton is a web of filaments that runs throughout the cytoplasm generally, and then in some cases into some of the other organelles, mainly the nucleus if we're talking about intermediate filaments. So there are three types of filament or fiber that you find in the cytoskeleton: microtubules, actin, intermediate filaments, and actin filaments. And these kind of, the name intermediate filament here, that name came from the observation that if you look at the diameter of these filaments, the actin filaments are the thinnest, the microtubules are the fattest, and the intermediate filaments, you guessed it, they're somewhere intermediate in diameter. So the name intermediate filament really comes from the diameter being intermediate, somewhere between that of actin filaments and microtubules. Actin filaments you'll sometimes hear described as microfilaments, and I'm going to try and stick with actin filament as the verbiage there. So let's start off with actin filaments and take a look at the structure of the actin filaments and see what's going on with the structure there. Okay, so here's an actin filament, and you've got to bear in mind this is a single actin filament and there's some kind of faded into the background. So these form a web, a network, a mesh-like structure. Now they do penetrate into the cytoplasm that kind of is deeper into the cell interior, but you find a lot of actin filaments just under the plasma membrane of cells, and they're going to help the cell, basically animal cells at least, move around and move extensions out from their cytoplasm so they can kind of crawl around. And then in other instances they're used for contraction, for example, in muscle cells. So what about the structure of the actin filament, also known as the microfilament, like we mentioned? Okay, so there are two strands of proteins that make up an actin filament. So one of the strands both of the strands are just actin proteins, individual folded actin proteins, and then you link those together. And so you take one actin filament protein, one actin monomer protein, you stick another one on, then another one, and another one. You get this long chain of actin protein monomers. So you got to think about this. Think about what happens. Different amino acids get linked together to make a polypeptide, which is then folded up to make an actin monomer, and then you stick a lot of those together like you'd stick pieces of Legos together to make a long chain. Then you take two of those chains and you twist them around one another, and you get then the actin filament. Now these filaments, they have discrete chemical ends. They have what's called a plus end, and they have a minus end, and you don't need to worry about the functionalities of those. You get in there in cell biology. So there's a little bit about actin filaments. You can add some context to that from your readings. Let's look next at the intermediate filaments. So these are the filaments which are kind of midway between actin filaments and microtubules in terms of diameter. Intermediate filaments are hugely diverse. If we look at actin filaments or microtubules, they're always the same structure and they're always made from the same proteins. Now, intermediate filaments, they have a general rule of thumb in terms of their structure, but there can be numerous proteins which are used to make up intermediate filaments. So probably the intermediate filament that's easiest for me to talk about is the lamin intermediate filament, and this is an intermediate filament which provides structure to the inside of the nucleus. So the nucleus is surrounded by two membranes, and then inside that inner membrane there is this network of these lamin proteins which give the nucleus some strength and some structure. So that's the easiest one for me to think about, and I'd recommend you maybe do a Google search on intermediate filament and see what other types of intermediate filament you can come up with. So what is the general structure here? You take two intermediate filament proteins and you twist them around one another like we saw just now, but then you kind of stagger those all together to make these bundles. And then when you get these big thick bundles of these overlapping staggered filaments, you get a structure which is incredibly strong. So these provide a lot of tensile strength to cells. And so you see these kind of all throughout the cell. You find them in the nucleus. Like I say, they're hugely diverse, very difficult to talk about. So do some reading online and see, just get a general idea of what's going on with the intermediate filaments. Okay, let's go out to the widest of the filaments. These are the microtubules, and these are the most structurally complex, if you like. So let's kind of look at the name to start with. They're a tubule. A tubule is a long, thin, hollow tube, basically. So it's a little narrow tube, which means if you look down the end of a microtubule, it wouldn't be solid. It'd be like looking down like a poster tube. It's hollow inside. So how do we build these microtubules? We build microtubules from two different protein monomers. So this is different than we saw in actin filaments, where there was only one protein monomer, and that was actin. Here we've got two protein monomers, alpha tubulin and beta tubulin. So what we do here is we take an alpha tubulin monomer and a beta tubulin monomer, and we stick two of those together, and that's called a dimer. Two proteins stuck together as a dimer. If the two proteins are different, that's called a heterodimer. If they're the same, it's a homodimer. So we get these heterodimers of an alpha tubulin and a beta tubulin. We stick those together, and then we add more and more and more of these dimers, and they slowly start to form these spiral-like structures as they kind of stack up, and you end up with this kind of, you can see it's kind of like a little spiral structure, if you like, with an inside called a lumen. The insides of things in biology are called lumens. So inside of the Golgi would be called the lumen. The inside of the endoplasmic reticulum would be called the lumen. The inside of the thylakoid membrane would be called the lumen. This just means the space inside. So the space inside here is called a lumen. Now these also have a plus and a minus end. And I'll tell you a little bit about the plus end. You don't need to worry about the minus end too much. The plus end, this is where the microtubule grows from. And so if we're adding on these dimers of alpha and beta tubulin, we're adding the dimers to the plus end. So these are very, very dynamic structures inside the cell. They're constantly growing out, and they shrink back, and they grow out, and they shrink back. And you look at the dynamics of that and the mathematics of that in cell biology in a bit more detail. The minus end in animal cells, they're anchored to one point in a cell, and that one point of anchorage is called the centrosome, and we'll look at those in a little bit when we look at some whole cells. So you have these minus ends anchored in the centrosome, and then the microtubule grows out from the plus end and shrinks and grows and shrinks and grows, and this growing and shrinking is called dynamic instability. So that's a little bit about the cytoskeleton. Like I say, supplement that with some of your own readings. So what I'm going to do now is I'm going to ask you a question. I'm going to put a question on the screen, and it's not really about the cytoskeleton. It's about those 4 levels of protein structure we've talked about. So you've got to think back to the 4 levels of protein structure. So here's the question. Let's see how you do with this. Okay, so let's talk this one through a little bit. What levels of protein structure are present in all filaments that make up the cytoskeleton? This is actually a little bit of a difficult question to answer because you've got to know something about the individual monomers. If we're dealing with a little monomer, let's deal with an actin. Let's deal with actin filaments to start with. We've got a monomer of actin protein. So there's a chain of amino acids. So that's primary structure. Now, if you looked at the actin monomer, you would see that it's folded up and there's secondary and there's tertiary structure. Then when we make the actin filament, we stick all of these actin monomers together, and that gives us quaternary structure. And you see the same thing in intermediate filaments in microtubules. So all levels are present. Now the thing that trips people up here is this quaternary structure. Don't forget that quaternary structure is one protein stuck on another. So if we do that and we add another and another and another, it's still quaternary structure. It's just getting more and more complicated. So, okay, let's look at where you find these structures inside the cell. Now this diagram is a little bit complicated, so let's talk about it for a couple of moments. On the left, we've got microtubules, but what are we actually looking at there? So is what someone's done is they've grown some cells in dishes, and you can do this in the lab. It's really easy. I have undergraduate students do this in my cell biology lab. You can grow dishes, cells, cultured cells, and these are probably cultured epithelial cells. You can grow them in dishes or on little microscope slides, and then you can basically stain different structures inside the cell. And so on the left, the microtubules have been stained. And when I say stained here, is what I mean is there's an antibody which will recognize the microtubules. So the microtubules are all throughout the cytoplasm in the cell, and its antibody comes along and recognizes the tubulin. And then we recognize the antibody with another antibody, and that antibody glows when you shine fluorescent light on it, and that lets us see where the microtubules are in the cell. So wherever you're seeing green in that left diagram, that's a microtubule, and you can see there are these thread-like structures, and they go all the way throughout the cytoplasm. The blue kind of oval in the middle towards the bottom there is the nucleus. And I'll point out the centrosome. It's a little bit difficult to see. The centrosomes are right here, and I know that because a lot of these microtubules trace back to this point here. The other thing that's worth noting here is that it looks like some of the microtubules are going directly through the nucleus, but you've got to remember, this is a three-dimensional cell, so it's sitting flat on a dish, but it's got this dimension, it's got Y dimensions, so it may be, let's say it's this thick, if we've zoomed in, a billion times, it's this thick, and the nucleus is sitting in the middle. Some of those microtubules are going over the top, and they're going underneath. So you've got to be careful, because it looks like the microtubules go through the nucleus, but they don't. You're looking at things going over the top or underneath in some cases. So those are the microtubules. Now the cool thing about microtubules is this dynamic behavior they exhibit. When we look at how cells divide, the microtubules are going to completely rearrange. So I want you to kind of keep this burned into your mind, because we're going to totally rearrange those microtubules to form some different structures to do cell division. In the middle here, it's the same thing. We've stained the intermediate filaments. Now you can see there that they're a little bit more kind of higgledy-piggledy. They're not as organized as the microtubules. The nucleus isn't stained here, but it's this structure here. Now the intermediate filaments, some of them tend to be associated with a lot of the Golgi in the endoplasmic reticulum. So you sometimes see them concentrated around the nucleus. Now here are the actin filaments. And so actin filaments sometimes bunch together to form what are called stress fibers, so that cells can resist pulling stresses. You can see some individual ones, some little individual ones here, and then you can see these regions where you've got this bright yellow. That's the actin filaments right around the edge of the membrane. So you're going to see a lot of that. So those are the actin filaments. The nucleus is kind of hidden under here, and again you can see how you've got some of the fibers going over the top of the nucleus there. So that's a little bit about the cytoskeleton. Let's just kind of review really quick, just to refresh your memory what we talked about so far. What proteins are microtubules made from? Okay, I'll put the answer into that next time. So, what we've talked about here really are two main things. We've talked about the cytosol and some of the functions of the cytosol and where you find the cytosol, and then we've talked about the cytoskeleton, the microtubules, the intermediate filaments, and the actin filaments. You want to support what I've said here with your readings, and I want you to be thinking about the relationship between structure and function. Okay, check out the learning outcomes. There's a lot of stuff in the learning outcomes to direct you to some additional readings, and I will catch up with you next time.